Hello everyone, my name is Dr. Syed Nomanata, and today we are briefly going to discuss different modalities for the treatment of hepatitis C. We will go through the structure of the hepatitis C virus, and then we will see how different drugs available act and where do they act. So let's jump right into it. So hepatitis C virus, it's a small virus belonging to the Flaviviridae family, and it's an enveloped virus. It consists of a lipid bilayer, a lipid membrane, which is very similar to a cell membrane. And in this lipid membrane, there are different envelope proteins which project as spikes from the surface of the viral particle. They include envelope proteins 1 and 2, and they are important in gaining entry within the cell, while the lipid membrane actually protects the genetic material of the virus. Now within the viral particle, we've got a protein core which is called capsid, and this protein core actually protects the genetic material of the virus, which is in the form of one long strand of RNA. As it is apparent in this slide that the genome of the HCV is actually protected within this capsid core. Now this is an oversimplified image of the HCV RNA. It's actually been simplified into one long strip. Now RNA consists of both coding and non-coding regions. Non-coding regions, they don't translate into proteins, whereas coding regions, they translate into proteins. As far as coding regions are concerned, there are two different types of coding regions. The first region codes for structural proteins, which make up the part of the viral particle, and the second part of the genome codes for non-structural proteins, which are not the part of the viral particle itself, but they are involved in translation, replication, and eventual assembly and export of the viral particle outside the hepatocyte. Now, as far as the structural proteins are concerned, there are three structural proteins which are encoded by the HCV genome, and they include the core protein, which makes up the capsid, and the envelope proteins E1 and E2, which are forming part of the viral envelope, and as explained earlier, they help the particle to gain entry within the cell. Again, this oversimplified image of the viral genome. Now we will see the different non-structural proteins. Now the non-structural proteins, there are quite a lot of them, and it is these proteins which are the target of the different antivirals, as we will see for in the upcoming slides. The first region of this genome codes for a protein called P7. It's a small protein which participates in viral assembly and its eventual release. Then comes the region which codes for non-structural protein 2, which along with multiple other viral proteins plays a key role again in viral assembly. The next region then codes for non-structural protein 3, or NS3. Now NS3 protein is actually an enzyme which has got both serine protease activity and helicase activity. Now the serine protease part actually cleaves different proteins which are once translated. Different proteins are not translated as isolated proteins. Instead, they form one long protein strip, which is then cleaved into individual protein segments. So the serine protease part actually cleaves this polyprotein into different proteins, and the helicase part actually is involved in unwinding of the viral RNA, and thus it supports the viral replication. Then we have got the non-structural protein 4A. Now this actually acts as a cofactor for non-structural protein 3A, and actually helps in its activity. So both NS3 and NS4A are very important for one another, and we will see that there are actually drugs which targets both of them at the same time. And then finally we've got non-structural protein 4B, which has got different functions important for viral replication. Then we've got another key protein, non-structural protein 5A, which interacts with other viral key proteins and host proteins to regulate the viral replication and assembly. It's a very key protein which is important for the viral survival. And finally we've got a non-structural protein 5B, which is actually the RNA dependent RNA polymerase, the most important enzyme which is critical for the viral replication. So NS5B is actually the enzyme which produces multiple copies of the viral RNA. Now, as far as the drugs are concerned, we've got direct acting oral agents which have revolutionized the treatment of hepatitis C. We've got ribavirin, which is again an oral agent, but the mechanisms are different as compared to the direct acting agents. And then finally we've got pegylated interferon alpha 2A, which is given in the form of weekly injections. Now, direct acting oral agents, we don't give them in isolation. They are usually given as part of a combination, as a synergistic combination which targets different parts of the viral genome, and that way they function more effectively, and there are lower chances of viral resistance as well. So the first direct acting viral combination is the combination of sofosbuvir and velpatasvir. Sofosbuvir targets the RNA polymerase non-structural protein 5B, whereas velpatasvir targets non-structural protein 5A. And as mentioned before, both of them are key players important in viral replication. Now, in this diagram and in the diagrams which will follow, these drugs do not actually target the RNA itself. This diagram is the diagram of the RNA and the different proteins that the RNA translates into, whereas the antiviral drugs actually target the actual protein, not the RNA. Then we've got this three-drug combination of sofosbuvir, velpatasvir, and voxilaprevir. Voxilaprevir additionally inhibits NS4A and NS3, which, as mentioned earlier, they act in combination with one another. So this three-drug combination is used for usually multi-drug resistant cases or cases which have failed earlier regimes. It's not given very commonly. It's not the first line regime. Then we've got sofosbuvir ladipasvir combination. Ladipasvir inhibits non-structural protein 5A, and sofosbuvir, as mentioned earlier, inhibits non-structural protein 5B, which is the RNA polymerase. Then we've got another drug combination by the name of elbasvir and grazoprevir. Now, grazoprevir inhibits non-structural protein 3 and 4A, whereas elbasvir inhibits a non-structural protein 5A. Then we've got glecaprevir and pibrentasvir. Now, pibrentasvir again inhibits non-structural protein 5A, and glycaprevir inhibits non-structural protein 3A and 4A. Now, again, just to summarize the key concept that the direct acting agents are present in the form of fixed drug combinations, and each drug actually inhibit a different non-structural protein target. So the combination is actually targeting different non-structural proteins, and that way they act synergistically to one another. And then we've got ribavirin. Now ribavirin is it's actually a compound which is structurally similar to guanosine, and once it goes inside the cell, it's phosphorylated just like any other nucleotide. So it can be phosphorylated into monoform, diform, or triform, and once converted into these phosphorylated forms, that's how it actually exerts its action. So in monophosphorylated form, it inhibits this enzyme which is called inosine monophosphate dehydrogenase, which is involved in replenishing a cell's GTP supplies, that is guanosine triphosphate supplies. So by inhibiting this enzyme, the GTP supplies of the cells, they start running low, and without GTP, a viral particle cannot replicate. Once it is converted into a triple phosphate form, it is able to inhibit the actual viral RNA polymerase, and that's how it reduces the actual replication process. Additionally, it can be incorporated into the viral genome because the structure is quite similar to guanosine. And once incorporated into the actual viral genome, it creates mutants because it's not an actual natural nucleotide. Therefore, in whichever genome it is incorporated, it does not function as normal. So essentially, you have got a non-functioning viral particle. And finally, it exerts its effect on the helper 2 cells, which we know have an important role in antibody formation that will come into play against the hepatitis C virus. And last but not the least, we've got pegylated interferon alpha 2A, which is derived from a human interferon. Interferon, we know, are natural antiviral compounds circulating in the bloodstream, and they play a key role in the innate immunity against the viruses especially. So once the molecule binds to cell surface receptors, it actually increases innate viral response through the JAK-STAT pathway, and that's how interferons function. So the mechanism of action is quite different from the other antiviral agents, as we have seen. That's all for today's lecture, guys. I hope you have enjoyed it, and we will meet again with a different topic soon. Take care.